Dear viewers, this video is part 2 in the 4-part series on genetic control of cell function in Medicine for Common Man series. Ribosomal RNA. The third type of RNA in the cell is ribosomal RNA, which constitutes about 60% of the ribosome. The remainder of the ribosome is protein, including about 75 types of proteins that are both structural proteins and enzymes needed to manufacture proteins. The ribosome is the physical structure in the cytoplasm on which proteins are actually synthesized. However, it always functions in association with the other two types of RNA. tRNA transports amino acids to the ribosome for incorporation into the developing protein, whereas mRNA provides the information necessary for sequencing the amino acids in proper order for each specific type of protein to be manufactured. Thus, the ribosome acts as a manufacturing plant in which the protein molecules are formed. So, dear listeners, by the way, this educational video is made possible by Prescribon, the makers of e-prescription software for digital prescriptions, available freely for doctors on the website www.prescribon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. Formation of ribosomes in the nucleolus. The DNA genes for the formation of ribosomal RNA are located in 5 pairs of chromosomes in the nucleus. Each of these chromosomes contains many duplicates of these particular genes because of the large amounts of ribosomal RNA required for cellular function. As the ribosomal RNA forms, it collects in the nucleolus, a specialized structure lying adjacent to the chromosomes. When large amounts of ribosomal RNA are being synthesized, as occurs in cells that manufacture large amounts of protein, the nucleolus is a large structure, whereas in cells that synthesize little protein, the nucleolus may not even be seen. Ribosomal RNA is specially processed in the nucleolus, where it binds with ribosomal proteins to form granular condensation products that are primordial substitutes of ribosomes. These subunits are then released from the nucleolus and transported through the large pores of the nuclear envelope to almost all parts of the cytoplasm. After the subunits enter the cytoplasm, they are assembled to form mature functional ribosomes. Therefore, proteins are formed in the cytoplasm of the cell, but not in the cell nucleus, because the nucleus does not contain mature ribosomes. miRNA and small interfering RNA. A fourth type of RNA in the cell is microRNA, miRNA. miRNAs are believed to play an important role in normal regulation of cell function, and alterations in miRNA function have been associated with diseases such as cancer and heart disease. Another type of miRNA is small interfering RNA, siRNA, also called silencing RNA or short interfering RNA. The siRNAs are short, double-stranded RNA molecules comprised of 20 to 25 nucleotides that interfere with expression of specific genes. siRNAs generally refer to synthetic miRNAs and can be administered to silence expression of specific genes. They are designed to avoid nuclear processing. After the siRNA enters the cytoplasm, it activates the RISC silencing complex, blocking the translation of miRNA. Because siRNAs can be tailored for any specific sequences in the gene, they can be used to block translation of any mRNA and therefore expression by any gene for which the nucleotide sequence is known. Researchers have proposed that siRNAs may become useful therapeutic tools to silence genes that contribute to the pathophysiology of disease. This figure 1.4 shown earlier shows the assembly parts of protein formation. Shown are the mRNA, a tape-like structure, ribosome, transfer RNA, and the protein assembly by amino acids, one amino acid at a time. Translation. Formation of proteins on the ribosomes. See figure 1.4, diagram of formation of protein. When a molecule of mRNA comes in contact with the ribosome, it travels through the ribosome, beginning at a predetermined end of the RNA molecule, specified by an appropriate sequence of RNA bases called the chain initiating codon. Then, as shown in figure 1.4, while the mRNA travels through the ribosome, a protein molecule is formed, a process called translation. Thus, the ribosome reads the codons of the mRNA in much the same way that a tape is read as it passes through the playback head of a tape recorder. Then, when a stop or chain terminating codon slips past the ribosome, the end of a protein molecule is signaled and the protein molecule is freed into the cytoplasm. Polyribosomes. A single mRNA molecule can form protein molecules in several ribosomes at the same time because the initial end of the RNA strand can pass to a successive ribosome as it leaves the first, as shown at the bottom left in figure 1.4 and figure 1.5. The protein molecules are in different stages of development in each ribosome. As a result, clusters of ribosomes frequently occur, with 3 to 10 ribosomes being attached to a single mRNA at the same time. These clusters are called polyribosomes. An mRNA can cause formation of a protein molecule in any ribosome. There is no specificity of ribosomes for given types of protein. The ribosome is simply the physical manufacturing plant in which the chemical reactions take place. This figure 1.5 shows the relationship of tRNA, mRNA, ribosomes, and endoplasmic reticulum. Many ribosomes attach to the endoplasmic reticulum. See figure 1.5 shown earlier. tRNA, mRNA, and ribosomes and endoplasmic reticulum. Any ribosomes become attached to the endoplasmic reticulum. This attachment occurs because the initial ends of many forming protein molecules have amino acid sequences that immediately attach to specific receptor sites on the endoplasmic reticulum, causing these molecules to penetrate the reticulum wall and enter the endoplasmic reticulum matrix. This process gives a granular appearance to the portions of the reticulum where proteins are being formed and are entering the matrix of the reticulum. Figure 1.5 shows the functional relationship of mRNA to the ribosomes and the manner in which the ribosomes attach to the membranes of the endoplasmic reticulum. Note the process of translation occurring in several ribosomes at the same time in response to the same strand of mRNA. Note also the newly forming polypeptide chain, that is protein chains, passing through the endoplasmic reticulum membrane into the endoplasmic matrix. It should be noted that except in glandular cells, in which large amounts of protein containing secretory vesicles are formed, most proteins synthesized by the ribosomes are released directly into the cytosol instead of into the endoplasmic reticulum. These proteins are enzymes and internal structural proteins of the cell. Figure 1.6 shows the formation of a protein molecule. First, the amino acid is activated, then it combines with the specific tRNA, then it forms part of the developing new protein. Chemical steps in protein synthesis. Figure 1.6 shown earlier, that is the formation of protein. Some of the chemical events that occur in the synthesis of a protein molecule are shown in figure 1.6. The stages of the reactions are as follows. 1. Each amino acid is activated by a chemical process in which ATP combines with the amino acid to form an adenosine monophosphate complex with the amino acid. The activated amino acid, having an excess of energy, then combines with its specific tRNA to form an amino acid tRNA complex. The tRNA carrying the amino acid complex then comes in contact with the mRNA molecule in the ribosome, where the anticodon of the tRNA attaches temporarily to its specific codon of the mRNA, thus lining up the amino acid in the appropriate sequence to form a protein molecule. Then, under the influence of the enzyme peptidyl transferase, one of the proteins in the ribosome, peptide bonds are formed between the successive amino acids, thus adding progressively to the protein chain. These chemical events require energy from high energy phosphate bonds. A total of 4 high energy bonds are used for each amino acid added to the protein chain. Thus, the synthesis of proteins is one of the most energy consuming processes of the cell. Synthesis of other substances in the cell. Many thousand protein enzymes formed in the manner just described control essentially all the other chemical reactions that take place in cells. These enzymes promote synthesis of lipids, glycogen, purines, pyrimidines, and hundreds of other substances. Control of gene function and biochemical activity in cells. It is clear that the genes control both the physical and chemical functions of the cells. However, the degree of activation of respective genes must also be controlled. Otherwise, some parts of the cell might overgrow or some chemical reactions might overact until they kill the cell. Each cell has powerful internal feedback control mechanisms that keep the various functional operations of the cell in step with one another. For each gene, that is about 20 to 25,000 genes in all, at least one such feedback mechanism exists. There are basically two methods whereby the biochemical activities in the cell are controlled. One, genetic regulation, in which the degree of activation of the genes and the formation of gene products are themselves controlled. And two, enzyme regulation, in which activity levels of already formed enzymes in the cell are controlled. Genetic regulation. Genetic regulation or regulation of gene expression covers the entire process from transcription of the genetic code in the nucleus to the formation of proteins in the cytoplasm. Regulation of gene expression provides all living organisms with the ability to respond to changes in their environment. In animals that have many different types of cells, tissues, and organs, differential regulation of gene expression also permits the different cell types in the body to each perform their specialized functions. Although a cardiac myocyte contains the same genetic code as a renal tubular epithelial cell, many genes are expressed in cardiac cells that are not expressed in renal tubular cells. The ultimate measure of gene expression is whether and how much of the gene products, that is proteins, are produced because proteins carry out cell functions, especially by the genes. Regulation of gene expression can occur at any point in the pathways of transcription, RNA processing, and translation. The promoter controls gene expression. Synthesis of cellular proteins is a complex process that starts with transcription of DNA into RNA. Transcription of DNA is controlled by regulatory elements found in the promoter of a gene. The basal promoter consists of a sequence of bases known as TATAAA, called the TATA box, the binding site for the TATA binding protein, and several other important transcription factors that are collectively referred to as the transcription factor IID complex. This basal promoter is found in all protein coding genes, and the polymerase must bind with this basal promoter before it can begin traveling along the DNA strand to synthesize RNA. The upstream promoter is located farther upstream from the transcription start site and contains several binding sites for positive or negative transcription factors that can affect transcription through interactions with proteins bound to the basal promoter. The structure and transcription factor binding sites in the upstream promoter vary from gene to gene to give rise to the different expression patterns of genes in different tissues. Transcription of genes is also influenced by enhancers, which are regions of DNA that can bind transcription factors. Enhancers can be located at a great distance from the gene they act on, or even on a different chromosome. They can also be located upstream or downstream of the gene that they regulate. Although enhancers may be located far from their target gene, they may be relatively close when DNA is coiled in the nucleus. It is estimated that there are more than 100,000 gene enhancer sequences in the human genome. In the organization of the chromosome, it is important to separate active genes that are being transcribed from genes that are repressed. This separation can be challenging because multiple genes may be located close together on the chromosome. The separation is achieved by chromosomal insulators. These insulators are gene sequences that provide a barrier so that a specific gene is isolated against transcriptional influences from surrounding genes. Insulators can vary greatly in their DNA sequences and the proteins that bind to them. So, dear viewers, this is the end of part 2 in the 4-part series on genetic control of cell function in Medicine for Common Man series. Thank you for watching.